My name is Muhammad Ali, currently a third-year student at Mount Sinai Medical School. On behalf of all the co-authors, I'll be presenting our work out of the Manning School of Biomedical Engineering at Cornell University on how VEGF signaling causes stalls in brain capillaries and reduces cerebral blood flow in Alzheimer's mice. ASL MRI reveals as much as 40% reduction in cerebral blood flow in Alzheimer's patients, measured in milliliters per 100 grams per minute, as demonstrated by the white arrows in the lower panel of the figure. Such reductions have also been recorded in mouse models of Alzheimer's disease. A large body of research suggests cerebral blood flow reductions likely contribute to cognitive impairment. However, the mechanisms driving cerebral blood flow reductions in Alzheimer's remain unclear. Recently, Hernandez and Bracco et al. Demonstrated that an increased incidence of stalled capillary blood flow caused by adhesions of leukocytes to the brain microvascular endothelium leads to 17% reduction of cerebral blood flow and exacerbates short-term memory loss in multiple mouse models of Alzheimer's disease. The illustration from the present manuscript shows a flowing capillary, where the blood cells indicated by a lack of Texas red labeled plasma move freely from one frame to the next over 5 seconds, as well as a stalled capillary where the blood cell remains stationary over 5 seconds. The fraction of capillaries with stalled blood flow is approximately 5 times greater in Alzheimer's mice than the wild-type mice. White blood cells, platelets, and red blood cells can all be present in stalled capillaries. However, white blood cells, specifically neutrophils, are present in approximately 70% of stalled capillaries. When stalled capillaries were released by the anti-Ly6G antibody against neutrophils, cerebral blood flow improved 17%, cognition improved, and amyloid burden decreased in Alzheimer's mice. However, anti-Ly6G leaves the mice neutropenic, making the therapy not compatible in Alzheimer's patients. In order to address stalled capillaries in Alzheimer's mice, we must probe what seeds capillary stalling. Could oxidative stress and vascular inflammation provide a nidus for stalling? Could the pathological breakdown of the neurovascular junction and local blood-brain barrier leakage provide a nidus for stalling? Are obesity, hypertension, traumatic brain injury, stroke, and the lack of exercise also contributing to capillary stalling? In the present study, we focus on how modulating blood-brain barrier regulation by inhibiting VEGF signaling may release capillary stalls and address brain blood reductions in Alzheimer's disease mice. Recent et al. Recently demonstrated that peripheral inhibition of VEGF receptor 2 improved clearance of microsphere-induced cortical capillary obstruction in wild-type mice, and a body of work demonstrates that VEGF levels are dysregulated in Alzheimer's disease patients. Our first experiment involved 4 cohorts of mice, 2 APPPS1 genotype and 2 wild-type genotype. In each of the 2 cohorts, treatment was given, either anti-VEGF injections or saline. The mice underwent craniotomies, and after 2 weeks of rest, were given treatments 3 separate days over a period of 1 week, followed by imaging, followed by another set of treatment and imaging again. The mice were then sacrificed, and their brains were used for ELISA and histology. Anti-VEGF-A antibody treatment reduced the number of stalled capillary in Alzheimer's disease mice by 3 to 4 times, bringing the fraction of stalled capillaries on par with wild-type mice. The anti-VEGF-A antibody concomitantly increased capillary speed in APPPS1 mice without a change in vessel diameter. Successive line scans were constructed into a space-time plot, where the x-axis represented distance along the vessel axis and a y-axis represented time. Diagonal stripes on this plot were formed by moving red blood cells and had a slope that was inversely proportional to red blood cell speed. The slope was shallower in the treated mice, indicating increased red blood cell speed. Anti-VEGF-A antibody also modulated endothelial protein expression in APPPS1 mice, reducing VEGF levels, eNOS levels, and increasing occludin levels, as can be demonstrated by the 4 panels in the middle of the illustration. Stalled capillaries also had a lower expression of occludin compared to flowing capillaries. The left illustration is live in vivo 2-photon microscopy, and the right image is histology of the same set of capillaries. The white line represents flowing capillaries and the yellow stalled capillaries. The illustration on the right demonstrates how the stalled capillary has lower occludin expression along its length as compared to the flowing capillaries. Anti-VEGF-A antibody treatment reduced capillary stalls and blood flow speed differences within 1 hour in the second experiment. After craniotomy, mice were given anti-VEGF treatment, and an hour after the treatment, stalled capillaries were released, as can be demonstrated by the 3 yellow stalled capillaries at baseline, all 3 of which were released at 1 hour after VEGF treatment. In a series of these mice, stalled capillaries were reduced 1 hour after injection and a week after injection, and there was a concomitant increase in capillary speed, whereas no such differences in capillary diameter were seen. This APPPS1 capillary can be seen with a reduced angle in the time-space plot at baseline, 1 hour, and 1 week, demonstrating increased red blood cell speed. Anti-VEGF-A treatment antibody reduced blood-brain barrier permeability within 1 hour in APPPS1 mice as well, in the cortex, but not in the hippocampus. Occludin levels were also reduced in brain samples of Alzheimer's disease patients. In summary, this study describes how increased peripheral VEGF-A and eNOS levels in the microvasculature, as well as reduced occludin concentrations at the neurovascular junction of APPPS1 mice, contributed to impaired blood-brain barrier integrity, which correlated with increased capillary stalling and reduced cerebral blood flow. Furthermore, it describes how non-functional pro-angiogenic VEGF-A signaling seen in AD could contribute to reduced cerebral blood flow, likely through capillary stalling. Drugs targeting peripheral VEGF signaling could be employed to 1. Derive indirect evidence for vascular dysfunction and leukocyte obstruction in human capillaries by tracking changes in brain blood flow as a diagnostic measure, and 2. Reduce cognitive impairment associated with brain blood flow reductions in AD patients as a therapeutic measure. Thank you for taking the time to listen to our presentation.